So we were having an interesting conversation in the hallway around how do we build the library, the public library, or the way to share all of this data. George, you've started with a public approach. Would you maybe comment on that, and others can chime in?
Well, first of all, why do we need it? I mean, part of it, we've kind of hinted at that if you find something that looks new, you need to make sure that it is new, and you need to have a lot of healthy genomes, healthy with respect to the particular disease you're talking about. They can't just be generally healthy. You need to know something about them. So you need to be able to share both their DNA and also their health status, because nobody's perfectly healthy. They all have little idiosyncrasies. And so to find a real control for your affected set, you need to, and it needs to be big. Now, sharing it has had problems of people wanting to control the cohort that they've spent government money or maybe private money on, and people wanting to protect privacy. And they've intertwined these and entangled these in ways that are really no longer sustainable. In particular, privacy is very, very hard to protect in an era of the internet where so many things, so almost anything can be re-identified at this point.
Jonathan, you have comments on the sharing piece?
So, first of all, I do want to emphasize, we really are in a tipping point. Daily and weekly, I get emails from two areas. One, the cancer clinician, mostly physician scientists who are first to use the technology, and they are sequencing genes, and they are changing therapies based on that information. So we're absolutely in a tipping point there. When I say a tipping point again, though, I do use words like physician scientists in these major centers. It's not the community hospital yet. And I do see a tipping point on major pediatric centers where people recognize, based on the examples you spoke about, that you can change somebody's life if you understand something early. So I see those things tipping. In terms of everything between, and how much we know now and how much we can know by having these large correlative studies or causative studies and having that shared, I do see huge obstacles. And some of it I actually think stems from the terrible state of funding right now in the biological sciences. My wife is a physician scientist at Yale, and this really is the worst period in decades in terms of the pay line, how you get that money. And as resources are scarce, for people to do the studies, whether it's a cancer study or an autism study or a study on schizophrenia, while the resources are scarce, each of those researchers is thinking about getting their next publication and having their own data for the next grant. And the world has changed. You really can't understand these complex diseases with 10 or 100 patients. And you do have to share. But I do see, and again, it's just bad timing. We have this beautiful technology, we're at these tipping points, and all of a sudden, there's not the money there, and researchers are fighting. So unfortunately, I'm not giving you an answer. I'm giving you a problem and a threat that I see every time I visit these centers, and it really hurts me. Yesterday, I was at Yale. I saw a beautiful presentation. They talked about the lung cancers they had sequenced. First hand went up, and it was, hey, how do I get access to the sequence? And they did not confront that directly. As George said, it gets mixed up in the answer. But the bottom line is, outside of George and maybe a few other individuals, we aren't having the sharing that we absolutely require to understand these complex diseases.
So if patients should own their genomes, how do we involve patients as a catalyst in breaking this barrier?
Well, before we do that, I think we can also have a discussion about a potential solution around this. And it's not just from the patient. But I will tell you, I have my genome right here. There's an app for that. It's called My Genome. You can get it for 99 cents. It gives you Jay Flatley's genome. My genome's not on there. But if you want to see my genome at a break, I'll show you my genome. That's not an effective way to share data. It's 3.2 billion data points. What we need to do is start asking some questions. And I think we can ask questions very different. We have kids that need your data. we can simply go ask the question, we have this variant that we're not sure about. Do you see that variant, yes or no? I think we can start in a much simpler environment of asking that question between institutions before we get to the point of putting everything in the public domain. I agree with George. I think that's helpful. But there's a lot of barriers around that. And I think we can share one data point at a time, and we can do this in a clinical environment that is safe and secure. And we can do that today. And then to your point that the patient has control. So I'll step back and let you go with that.
Well, actually, it's a good thread. So, Steve, would you, in your endeavors in sequencing the genomes that you're about to sequence, be willing to consider an approach like that?
Yeah, absolutely. And I think we go back to our fundamental premise in our whole business, which is that we're here to serve the patient. And we don't have any records. They're all patient records. And the genome belongs to the patient. And if the patient wants to share it with anyone else, they should have every right to share it with everyone else. I think also we struggle to have the right conversation on these issues, and that there isn't enough money to be able to advance this premise is really bizarre. I mean, if you look at the total value equation and you look at the economic potential and the human potential that lies here. And just in oncology, we were talking in the green room, we're going to spend $80 billion on cancer care in the US this year. And if you go back to the way the system works, and the double-blinded studies and applying therapy to everyone, we know that a very large component of that is not going to be successful. And the fact that we can't work across all these lines with the FDA, with the insurance companies, with the employers, with the patients, with the providers, and find some ways to accelerate this and break through it is really a strange expression of innovation in America.
And the other part that I think is important is that, does it really make a difference? So we've moved diagnosis from between 5 and 10% of the time to 34% of the time with whole genome sequencing. And our limitation is we don't have the ability to compare more genomes. We use George's personalized genome project. We use those data. We use everything we can get our hands on. But just imagine how far can we move that as we share these types of data. I mean, it's doable.
And how many is enough?
I'm going to give George the answer because I think I agree with him.
More is better. 1 million would be great. 7 billion would be perfect.
I think that's the right number, too.
But all for N of 1 application, right?
That's right. You need to have 7 billion, but you can still study and get causative information from as little as 1. Nick Poulter was essentially a N equals 1, and there are many other examples like that, and the number is growing. I think that it's really a golden age for this sort of thing, and sharing is almost inevitable to some extent. It's a big change in paradigm that patients get their data now. When we started our personal genome project, that wasn't really very commonly practiced. Once they have their data, then they're very often willing to share it because they want to save families like theirs around the world, and they're highly motivated to do that. And so many of the genomes that we post on the internet are ones that we didn't actually sequence. They got it some other way.
I think this N of 1 is certainly an interesting concept as we think about some of the conversations earlier today about how we do clinical trials. They're certainly not an N of 1. So how do you see that unfolding in the future in terms of impact on both drug development and approval processes?
So I think there's two ways to look at it. I'll do an N of 1 experiment. I had shingles a month ago. They put me on all kinds of new medications. I hate taking new medications. I'm a pharmacologist. So I went to PharmGKB. I pulled up what the variants are for those drugs. I went to my genome. I didn't have any of those variants, but I do have variants, okay? I took the drugs. I'm still here, okay? That was an experiment, right? That was an N of 1. So we just captured that those variations, at least in me, are meaningful. Every time you take a medication and it works and you don't have a problem, that's an experiment. So how do we capture that?
Facebook?
I didn't. I should do that. But I'm serious. I mean, so every time we do this, we are doing an experiment. It's not quite the randomized clinical trial, but the randomized clinical trial says on average, I should be fine taking the drug. When I take it, it is in practice a trial.
Yeah. I would say what we're really talking about here is phase 4 clinical trials. It's basically once it's out there in the population. And we're not asking whether a drug is good or bad in phase 4. We're asking whether there are environmental and genetic components that mitigate or streamline or target certain populations or exclude them. And I think that's extremely important. And you can call it whatever N you want, but in the end, each person has their own unique set of environmental and genetic components.
But even randomized clinical trials operate off the assumption that you control the variables and can see whether one variable makes a difference or not. And at the genomic level, I just don't think that's realistic. It's not the way it works. We're all different. We're selecting two, and in the case of cancer, it's where the cancer presented itself first, so that's one. But underneath it, you have all these uncontrolled variables, and you apply one, and then you apply it to a whole population, and you get some response, and from that, the whole industry responds. So I think we're really operating with a fallacy anyway. And I'm not saying we can't and shouldn't pay attention to efficacy and safety. We have
Absolutely.
to figure that out. But I don't think we can hide behind large population randomized clinical trials anymore. It's got to be a better way, as Andy von Eschenbach was pushing us.